Thank you for the introduction, Tia. Thank you for the organizers. Thank you to the organizers for giving me the opportunity to present this talk to you. I'm going to briefly talk about, well, a couple of custom-tailored Galaxy tools for plant immunity and pathogen informatics. And, well, this is one of the first talks of the now renamed institute, which was formerly known as TGAC, now known as Leibniz-Institut, and of course I managed to crop the logo wrong. So, why plant pathogen informatics? Well, we're eating a lot of plants. If you have these calculations from the Genome Center in the UK, is each and every one of us, and on conference days even more, eat about 50 wheat plants, 3 rice plants, and a half maize plants every single day. And if you look, for example, in wheat production, we see this blue curve here. So the wheat production is currently in decline, whereas the utilization is rising more and more with increasing population. The bar graph that we can see here is the stocks, which is currently, I think it's sufficient to get us over the winter for about 3 months or something. So if anything happens, we are the problems. And as if that's not enough, we have declining yields due to climate change, for example. We also have these fellas here. And we could see this with a recent outbreak in Bangladesh, where we have wheat blast, deadly wheat fungus, basically devastating field after field after field. And basically we need to do something because we have an average of 15% annual yield loss on top of already declining yield. And in susceptible variants, we have up to 70% yield loss. So where I'm coming from, in the United Kingdom, we have a little town called Norwich, where a couple of institutes, a couple of scientists across institutes are working on A, making plants more resistant, and B, understanding how the pathogens, how they work, how they attack, how to possibly prevent that to happen. So we have the Leibniz-Institut, huge computing center, lots of bioinformaticians there. We have DSL, small elite institute, working on mostly fungal plant interactions. University of East Anglia, which is the mother institution of TSL, and the Genome Center, which should be a world-known agricultural research institute. So in plant immunity and plant pathogen interactions, you can go 2 ways. Basically, either you look at the plants here and study their defense mechanisms. For example, you do comparative sequence analysis, you do motif analysis, structural studies to look where, for example, certain immune receptors, how they are structured, how they are built, to see where they are located, and then do maybe some crossing experiments or something to make other strains more resistant. You can do this with SNP analysis, indel analysis, and so-called SNP mining operations. On the other side, we have the fungi, but we also have other crop pests, such as not carnivorous plants, but some what are they called? Parasitic plants, where you can study the mechanism of the infestation, of how they actually bypass the host defense, how they reproduce. And this is, for example, done with an effector protein analysis. Effectors are basically the assault weapons of the fungal community. So usually whatever you do in bioinformatics or in general research there is, in bioinformatics you don't infect the plants, but in general research, in general plant immunity research, you somehow infect the plant with a pathogen, you observe, then sample and select. This is always part of the process. Generate huge amounts of data, the bioinformaticians have then play with that. So the first tool, what I'm going to talk about, goes into the direction of studying the defense mechanisms. So in plant immunity is a case of pattern recognition. So we have the gnome, one of the very widely distributed mechanisms is the direct pathogen recognition. Basically, we have the little pathogen here. We have an immune receptor protein. Notice the pathogen is there, so we can start some immune response. We have an indirect target recognition. Basically, the plant receptor, the immune receptor, is not monitoring for the presence of the pathogen, but it actually looks at where the pathogen attacks and then sees whether something is wrong there, and then triggers an immune response. And then there's this third mechanism, which is rather new, has been identified in rice and Arabidopsis. It's an integrated domain. Basically, we have co-evolving immune receptors which have an integrated sensor domain, which then can have a more efficient way of recognizing the presence of the pathogen, but they are not failing to recognize, for example, if the target has evolved further and has a mutation. So, and what my group did before I joined it, they made a study and found evidence of these integrated domain fusions, as we call it, across 49 plant species and over 14 families. So this is basically, this has been published in BMC Biology rather recently, and basically what we have here is a collection of Perl scripts. So we start with data from phytosome, we run Hammer, we're using the typical PFAM domain, parse the domains, then do something called domain fishing, do something called the NLR classification, which is looking for immune receptor domains, and then sees what kind of domains are overlapping there. So, as I mentioned, this is a Perl script collection, and it was developed for this systematic analysis, but we wanted to have this basically to provide this to scientists who want to see whether these integrated domains are present in their plants, which we did not cover. And so we built a Galaxy workflow for that with a couple of customized tools. So you take your plant proteome, you take idFusion, which either uses PFAM scan Perl or direct HMM scan results, then use a Python script-based tool, the idFusion parser, and get your outputs. Then you can identify potential domain fusions in the protein set. So what we are planning is also to reconstruct this systematic research so that you can compare multiple runs on multiple plants and then see more whether we can find it in even further widespread. So the second part of my talk is goes to the attack mechanisms of the pathogens of the effector protein analysis. And here we are hijacking metagenome informatics. When I think about metagenome, I mean, we have heard a lot, so I kind of skip the introduction, but usually we see, okay, we have this hot spring, we have the mouth that Daniel just told us about. And usually in metagenomics, we're looking at a multitude of microorganisms. In our case, we are not. So what we are doing is we have one host usually, and we are expecting 1 to 2 pathogen species, depending on whether we look on an extra interaction, and then maybe some contamination noise. And then in that regard, we can also say metagenomics or a contaminated sample of any kind is also some kind of metagenomic. For these effector protein mining, which involves, for example, transcript assembly and further downstream analysis, we definitely need pure samples, so we have to find some way to remove the host from the pathogen data, and, well, this is what I'm aiming at here with this tool. So what we usually do to clean these samples is we have to read data. When we have some kind of classifier, we can use BLAST, we can use short read mappers against some customized libraries. We can use metagenomic classifiers. Then we have the output, when we do some post-processing with that, and then we have to somehow get all the reads which have been classified as pathogen or also the unidentified ones, because we don't know to which they belong, and call these our clean data sets. So what we're using is the Kraken, as Daniel just explained. Thank you very much, because I don't like to explain how it works. So my boss, that was something I was working on last year. My boss said basically, okay, we need this to be wrapped in Galaxy, so go ahead. I've never worked on a tool before, never wrapped a tool, so first thing, wrap the Kraken. And I did it, and I checked. I looked in the tool shed, and it was completely empty, and I was super happy, so I'm the first one, yes, yeah, great. And then I just let it slide for a couple of months. And then there was this guy, Eric, who had wrapped it in the meantime, so I could never, so my first Kraken wrapper, basically, I was beaten by Eric. So thank you, you don't even have, not just have better hair than me, you're the king. So, well, but I have something that the Kraken wrapper does not have. So, well, so to use the Kraken classification results that we usually get and to use them for our purposes, we need some post-processing in order to make sense out of it. Because for us, it's not enough to just have a table and to visualize what we find, because we actually have to do something with what we find. So, and I have a little suite of tools. What's this suite? What's this? Just got them also. These are 3 Python-based tools: KT-extract, KT-combine, and KT-summarize. So KT-extract basically takes the read data that you just classified with your Kraken database, with your Kraken classifier, takes a taxonomy ID as the root of a subtree that you want to pull out. So for example, if I have fungal infection, I select I want to have all fungal reads, and it will extract me all fungal reads. I will have that as a new data set in Galaxy and can use this for downstream analysis. I can also exclude certain subtrees, or I can say I want all the unclassified or not, and so on. The second tool is KT-combine. So this is what we use if we are not completely sure. If we have these, this is specific, this is custom tailored for these 2 organisms in one metagenomic sample approach. Basically, we take one database which is specific for organism A, so for example for plants, one which is for B, for fungi, and then run Kraken classification on both, take the output, run Ktoolu on that, and then basically split the ones which are only classified as plants, only as fungi, then the ones which are shared, which can happen in some stuff like actin, for example, and then we take the unclassified reads. And that way we have the possibility to say we take only fungal classified reads, but we can also take the unclassified, which might have not been classified because they're just no sequences in the database yet. Okay, the third tool that we have is KT-summarize, and that is basically that does some statistics on the Kraken output, but also writes it into a format that we can use Krona tools visualization. And then it directly calls Krona tools, so we can we can see how our sample looks like. And this is how it typically looks like. This is unclassified in our samples, and we have a little bit of pathogen here. So to summarize is we have this is available at GitHub. Not yet in the tool shed, but I'm going to, if I have time to work on that, I will do that. We have used it successfully in plant-fungus interaction. We have used it successfully outside of Galaxy and plant-virus interactions. So that's also a thing that they can be used on the command line without having to have the Galaxy environment for people who can, who just don't have the possibility to run it on Galaxy. And what we are currently planning, this is together with one guy from my last lab, with Martin Page from TSL Bioinformatics. So what we are planning to do is we're going to hijack, so we're going to take the visualization, possibly hijacking the Ktoolu code or writing our own. And then you're basically doing something like you just select what reads you want directly from the visualization interface, and you get this stuff as Galaxy data sets in the background. That is what we are hoping to achieve at some point in the near future. So in summary, our food supply is in danger as plant pathogens can cause heavy yield losses. And there are some custom-tailored Galaxy-based bioinformatics solutions to help, hopefully, combating that successfully somehow. So we have idFusion to see whether there are some domain fusions. We have Ktoolu for effector mining, for example. Then I have two more pipelines to work for, or tools, which I couldn't talk about here. We have Snippler. It's the SNP mining data compilation tool for bulk segregant analysis, basically to see on the plant side, to check whether there can be some mutations correlated with pathogen resistance. And we have SpoutP, which is an effector mining-based workflow, post-processing analysis after the reclassification, after transcripts of assembly of the classified reads. So if anybody is interested in those, you know how and where to contact me, I hope, and then we can talk about that. So with that, I have some acknowledgements to make. So I'm thanking a couple of people here, my new boss, my old boss, very valuable member of the team, Martin Page, very good programmer, also the author of the Galaxy flavor generator. So Galaxy team hired that guy. TSO2 blades are plant fungus infections. TSL Jones lab are for OMIC interactions, which are some kind of fungi. Pirasteh Pahlavan was plant virus interactions. Big thanks to the business support, otherwise I wouldn't be here properly. And then a big thanks to the organizers. Really, really awesome. I mean, we started with an awesome hackathon thing, didn't have to leave the venue at like 5 or whatever it was last year. So, Indiana University team, great thing to do, you rule. So, thank you for your attention.
So, do you know on which system they validated your Kraken?
I'm sorry?
Which system on which plant fungus system did they validate the Kraken?
Cannot talk about that.
Huh?
It's classified.
Oh.
Sorry.
I have another question that may be a little bit silly. Would it be easy to adapt the Kraken software for another system, for example, soil, microbial communities where you have a lot of other contaminants?
You mean the Ktoolu?
Yeah, the same pipeline.
That doesn't matter. It's not.
Okay.
It's developed for plant something interaction, but it could easily be used for anything else. I mean, the only thing is that we can assume that every time we find a plant read, it's our host, because plant sequences are not that comprehensive as other, let's say, some more model organisms. So basically, we always find Arabidopsis somewhere because we just don't have the certain reference sequence for a couple of proteins or a couple of genes. So we have to assume that every plant read that we find, that's either the host, that we hope that's or somebody didn't clean the sequencer properly.
Thank you.